I realized the camera in my office is zoomed in a lot farther than the one in my home office. It's like I gotta sit back farther.
That's funny. It's amazing how much I started caring about what's going on back there, my walls and things. Never really thought about it before.
That is a handy thing with the Zoom backgrounds. It can
Yeah,
hide
for
the
sure. I guess we'll give folks a few more minutes to join, and then we'll get rolling.
Sounds good.
Yeah, we're getting up there. There's Vito. Just another minute or two. We got people still joining. So before I introduce Cole, I will remind everyone or anyone who has not been to one of these seminars before that this is a webinar format, and there is no direct, you can't ask the question directly, so we ask that you post your question in the chat, and either Kavi or myself will pose the question to Cole so he can answer it. And Cole, if there are good stopping points during the presentation, we can, it might be worthwhile to allow us to pose those questions. So otherwise, would you prefer us to just chime in when we get a chance, or I can't remember whether you should be able to see if a question pops up, I believe.
Okay, sounds good. I'm happy to be interrupted. It doesn't really matter to me.
Excellent. So I think I'm going to start introducing Cole. He is a third-year postdoctoral fellow in Bill Holmes's lab. He's been here for and been attending these QSBC seminars for a while. In fact, presented a couple years ago, and so we're honored to have him join us today. He's a card-carrying mathematician. He's got his BSc from University of Alberta and a master's in applied mathematics, both in Canada, Alberta, Canada. And then he went on to get a PhD in the University of British Columbia in Vancouver, also in mathematics. So I am always in awe of the true mathematicians that can help me understand this complex math. So he's going to talk to us today about mechanosensing and migration, and trying to interpret how cells are sensing our environment. Thank you, Cole.
Great. Thanks, Darren. Just going to go ahead and share my screen here. All right. So yeah, thank you for the introduction. Today I'm going to talk about mechanosensing and how it can enhance adaptation to maintain polarity in migrating cells. In particular, here's a little photograph of a teaser of one of the results that'll come up later, so you can look forward to understanding what I'm trying to show you here. But I've got some videos as well of these cool little migrating cells. Anyways, my name's Cole Zamerchuk. I'm a postdoc in the Physics and Astronomy department. I work with Bill Holmes, and the talk today is joint work actually with Bill Holmes and two biomedical engineers, Jared Collette, who's a graduate student and sort of the co-primary author on our recent publication, and Jared's mentor, Vijay Rajagopal. And so they have a cell mechanobiology and structure lab in Melbourne. So the motivation for the talk is this well, probably overplayed and very famous video, which was actually taken at Vanderbilt Universities in the 1950s. There's a little bit of a story about it, at this embryology University of New South Wales website, if you're curious and want to check that out later. But what I'm going to show you is a video of a neutrophil. It's a white blood cell that's chasing this little bacteria that's been added to this blood smear. And you can watch it migrate and change directions and organize itself into having a front and a back and bumping into the other red blood cells in the environment and kind of basically chasing the bacteria around. So yeah, I'm sure that many of you have seen this, and I don't know, I keep coming back to it because first of all, it's really fun to be part of the people that are showing the classic video, but it's also fun because I think it's a really cool video and you can sort of root on this neutrophil as it eventually envelops and grabs its little bacteria. Anyways, what I want to talk about is that this cell, as it's migrating and moving around in its environment, it's encountering a lot of variables. And in particular, it's encountering different chemoattractants, chemicals and other things in the environment that it responds to and then follows. And the cell has to make decisions about what to do and which way to move and how to move up gradients of chemoattractant and things like that. And so just by the way of background, I think most people in the audience here will know a lot more cell biology than me, but let me remind you that the movement and shape of kind of one of these cells is controlled by like the cytoskeleton, and in particular the actin cytoskeleton. The actin cytoskeleton is built up of small protein filaments, right? Actin filaments that polymerize and grow, and the cell is able to you know, attain a very a polarized shape where it can polymerize actin at sort of the leading edge and push itself forward. And it can also modulate the cytoskeleton, the actin cytoskeleton at the same time by you know, turning on molecular machinery like myosin, which are, you know, molecular motors that bind to these filaments and move them around. Okay, and so provided the cell is sticking to something at the bottom, it's able to basically push its front forward and pull its rear back towards it, thus inducing movement and migration. Anyways, this picture is from another one of these really famous papers from Svitkina, Journal of Cell Biology. Some great, cool microscopy images of the actin network. And you can see it's this dense, very filamentous material almost, that has some sort of physical and chemical properties. So in turn, there's a set of proteins that I've studied a lot in my research called the Rho GTPases, and they are by and large responsible to modulate the actin cytoskeleton. And these proteins, RAC, CDC42, and Rho, act as kind of central regulators of this big sort of black box signaling circuit. So they exist in an active membrane-bound form and an inactive freely diffusing form, which is important. So basically you can think of them as just a little molecular light switch. When they're on, right, they're signaling their downstream effectors. And when they're off, they are just kind of floating around or sequestered near the cell membrane. And the takeaway message is that, you know, they don't just turn on and off themselves. They seem to be influenced by other signals from the environment. So for example, you can look at this chart and see the G protein coupled receptors up at the top right, or you can see the receptor tyrosine kinases or integrin receptors. And in the middle, those RAC and Rho and CDC42 proteins. But they're the proteins that we focus our study on, and we try to understand how sort of the underlying signaling behaviors of RAC and Rho proteins affect the cell behavior. So one piece of cool evidence, and for those who were at my talk a couple years ago in this seminar probably remember this slide, but it's not just other sort of proteins and chemical signals that can affect signaling of RAC. For example, mechanical stimuli are also transduced and can enter that big signaling network and modulate RAC activity. So if they can modulate Rac activity, then mechanics can also then start to modulate the actin cytoskeleton. There's basically a big feedback loop between signaling and mechanics. And so this is another very cool result. I like to tell this story of this poor neutrophil. It's sitting on a plate somewhere, and this group, it's the group of Orion Weiner in California, and they take a micropipette and they stick it on to the neutrophil. So you can see the micropipette there labeled with a blue arrow, and the bright polarized Rac activity. It's localized to one side of the cell, and it's polarized, right? There's a high level in one area, and there's a low level in the other. And what they do is they basically turn on a mini vacuum cleaner and increase suction onto the cell. And what they notice very quickly is that the Rac polarized signal suddenly disappears. And so this work and the work of others has posited that membrane tension, or an increase in tension on the cell membrane or in the cell cortex, can indeed modulate signaling. And what I think is really cool is when they turn the aspiration off, in the figure on the right, they basically turn their vacuum cleaner off and the Rac signal returns. Okay, so the takeaway is that we're completing a circuit. Protein signaling like Rac can modulate actin, which can modulate cell behavior and movement. Cell behavior and movement and in interactions with the environment can modulate things like membrane tension or forces, and those forces feed back into the signaling circuit. And so there's a dynamic interplay between the two things that can play a role in cell migration. So another observation that came out of Orion Weiner's group, this one a little bit earlier, is an experiment where the cell is not in an environment with like a chemoattractant gradient, like it was in that video where it was probably chasing that little bacteria and the chemicals that the bacteria was interacting with or excreting. And so instead, what this neutrophil, this experimental assay did is they have one of these resting cells that's sitting there kind of at minus 24 seconds. And at t=0, what they're doing is they're dumping a high level of chemoattractant all over the cell. So you can think about it as this kind of very high level of uniform stimulation. And what they see is the Rac activity. This is a downstream readout, I'm not exactly sure what, fires up all throughout the cell. The cell becomes very highly active. And therefore, over the remaining sort of 30 or 40 seconds, the cell generates a few sites of Rac activity that kind of compete to be the new polarized site. But in the end, the cell is able to resolve this sort of oversaturating stimulus and end up with a single polarized front. Okay, I'll play the movie for you here so you can watch it in real time. But they douse it with chemoattractant, Rac fires up, and you can start to see these little polarity sites kind of compete until one fizzles out and the new polarity site at the top of the cell is sort of formed. Okay, it'll loop through once more. Right, and so our basic question in this project is how does this cell adapt to this high oversaturating level of stimulus? How is it able to maintain its polarity given this very high level of stimulus? So let me take you back in time to sort of the origins of this idea of biochemical adaptation. And another nice sort of very cool what we would consider low-tech paper, but I guess is state of the art to get into nature at the time, is a paper by Sally Zygmunt, where they took, again, neutrophils, and they placed them in a chemoattractant gradient and observed their behavior and their migration over time. And instead of capturing digital photography images, they sketched out the trace of the cells and then connected sort of the center points of each of the cell traces over time. And this is the paper that the figure that I copied from their paper. But you can see that the cells migrate preferentially up this gradient of chemoattractant, and they're able to figure out how to go that direction, right? They're able to somehow, and I can quote from the paper, there's some sort of time and concentration dependent threshold stimulation at the membrane which when activated leads to local pseudopod formation, meaning like a protrusive foot at the front of the cell, and some sort of refractory period inhibiting the pseudopod formation on the other positions. Meaning that the cell probably has some mechanism by which it's able to generate a new polarity front, and that polarity front will somehow have to inhibit other polarity fronts from forming. In this way, the cell is able to maintain its polarity and migrate up the gradient. Another mechanism that could work, and this was posited back in 1974, was that the cell could have some sort of internal computer, and it's able to compare the amount of stimuli that it gets sort of all around its periphery to determine which direction it needs to go. And so this takes us to the LEGI model, the local excitation global inhibition model. And I don't want to spend too much time on this, but I just want to share that there's been a lot of sort of both theoretical and experimental work looking into cell migration in response to chemoattractant gradients. And sort of the famous model that came out of this work is this LEGI idea. You can check out papers by like Pablo Iglesias and Peter Devriotis. They're the kind of two big names in the mathematical side of these LEGI models. But the basic story is this, is that the signal, like from the chemoattractant, is responsible for turning on two things. First, an excitatory variable, a local variable that gets excited. So wherever there's a signal, you have some excitation. At the same time, however, the second thing that the signal does is fires up an inhibitor of some kind. And this inhibitor is able to travel fast throughout the cell, or it's able to be measured in an average quantity all throughout the cell. It's got some sort of average global behavior. The excitatory variable is responsible for stimulating the response, like firing up the machinery to cause actin to polymerize and the cell to migrate. And the inhibitory variable is responsible for turning that response off. And so I borrowed a figure from this paper by Iglesias and Devriotis. And in this figure on the bottom left, they cartooned out sort of the response of the LEGI model to sort of a uniform stimulus. So what would happen in one of these cells if you were to apply a high level of chemoattractant? Well, all around the cell, you get excited. And what you can see in the curves is that the excitation levels at the front or the back of the cell would be the same. As a result of the signal, the inhibitory variable would also start to pick up and grow. Okay? And so the response at the front and the back basically would follow the same trajectory, right? They both spike, and then due to the inhibition that happens a little bit slower, or on average, that response is dashed. And so the cell ends up, well, kind of right where it started in this resting state. But in a gradient, and that's what the figure on the bottom right is trying to show, there's a distinct response at the front and the back. The stuff at the front is shown in solid lines, and the response at the back is shown with dashed lines. So in a gradient, where you have higher chemoattractant or higher signal at the front of the cell, you end up with a relatively high excitatory variable compared to the excitation at the back. This leads to a larger peak in the response variable at the front than in the back. And again, as the inhibition slowly climbs over time and affects both the front and the back equally, there becomes some sort of difference between the response variables at the front and the back. And so in this way, the cell can figure out, where's the front and where's the back. So that's a tour of kind of the basic biochemical models. And what we're proposing here to explain the observations from that 2007 Orion Weiner paper, where they have the high levels of chemoattractant dousing the cell, is that mechanics and other changes that involve the cell membrane and tension can serve as this long-range inhibitory feedback that allow the cells to adapt to the high concentration signaling environments. Okay, and so this is basically the sort of framework, is we have one of these resting cells, we douse it with a whole bunch of chemoattractant, the cell's Rac activity fires up, it starts to expand, and as it starts to expand, membrane tension or cell cortex tension builds up. And these mechanical effects then feed back into the signaling, causing the cell to adopt this polarized Rac activity and polarized shape. So I think this would be an okay spot to stop and ask if there's any sort of questions about the background or the experiments or the ideas, because next I'm going to move into sort of the results of modeling this process in 1D and 2D cells. All right, well, please feel free to interrupt me if a question does pop up. We're going to get a little bit into some of the mathematical details and trying to understand how we can model Rac signaling in these cells, how we can understand the adaptive response, and then I'll show you some results from some computer simulations which sort of play with our simulated cells in different scenarios to illustrate how this mechanosensing adaptation hypothesis can work.
So Cole, I can ask a quick question. The
Sure.
math that you were showing there, was that assuming essentially either a circular or a spherical shape of a cell? Do you know if that mathematical formula would have any requirement of the shape of the cell for it to?
You mean this kind of thing?
When you showed the temporal, the dynamics, that part. Does that?
Yeah. Well, yeah, so the shape I think is going to have play an important role, and that's an important thing. You know, by and large, this is just kind of trying to cartoon what would happen. It doesn't need to have a circular shape, and as far as I know, this Legge model kind of explains things in a whole host of cells. Mostly they think about dictyostelium migration and stuff like that, and so I think, you know, they have a whole array of morphologies when they think about this model. But yeah, no, I don't believe it depends on the circularity of this.
Excellent, thanks.
Okay. Right, so let me get going with the Rac signaling model. What I've done is adapted something known as the wave pinning model for Rac activity. So this consists of a very simple description of that big signaling circuit that I had up on a slide in the introduction. And here I've distilled all of the signaling interactions down to something very simple. Rac switches between an active and inactive form from the cytosolic to the membrane bound compartments. When it's membrane bound, it doesn't move around as much as it does when it's freely diffusing in the cytosol, just because the diffusion is slower in the membrane than in the cytosol. And the sort of only nonlinear signal that we input into the model is auto activation, so that when Rac is on, you can think of active Rac signaling other effectors, which in turn enhance its activation. Okay, and so this is kind of the simplest description of the well understood signaling kinetics that exist in that sort of circuit diagram for signaling that I showed before. And how these equations manifest is a set of partial differential equations which describe how the amount of Rac, that's R, and inactive Rac, that's R sub I, change in time and space. Okay, I'm just going to walk you through the terms here. The first thing that you see in the Rac equation here is this term in parentheses, which is the activation rate, of which inactive Rac enters the active Rac compartment. It has a basal activation rate B, as well as this auto activation response function with magnitude C. So this is like a little hill function. You know, it goes up and does something like that. Right? It's when Rac is sufficient amplitude, we increase the activation rate from B to B plus C. Okay. Rac also turns off with some rate delta. And this term here at the end is the diffusion operator, which describes how Rac diffuses from areas of high concentration to low concentration. Okay, in the inactive equation is essentially the mirror image of the active one because, as I said, Rac just switches on and off. So we have a minus sign in front of this term here, because this amount of inactive rac is leaving this compartment and becoming activated. And similarly, we have a plus sign here in this delta R term, because that amount of active rac is turning off. Okay. Lastly, there's also this diffusion term for the inactive rac with a different diffusion coefficient, DI. And how I'm kind of thinking about this applying to one of these cells is just initially is kind of like a 1D domain, where I'm thinking about like a cross section across the cell, and maybe the cell bottom or the part of the cell that's attached or adhering to the substrate. And I'm going to look for polarized patterns, regions where we have high activity in one side and no activity in the other. So just to explain, first of all, for the math aficionados in the room, one feature of this model, this sort of simple circuit, is the presence of bistability, meaning that it's possible for a single set of parameters to have a stable high rac state and a low rac state. And when you usually have a bistable system in the presence of diffusion, what's commonly known is you get traveling wave solutions, which is shown on the cartoon that I stole from one of Bill's paper on the left. So you have an initial perturbation starting at the left-hand side of the cell, or in your domain. This is commonly used in ecology a lot. And this wave propagates to the right with some speed. Okay. Now the difference between that bistable wave that travels all throughout the domain and what we have in the cell polarity model is that the wave becomes pinned. So it starts out in the same way, but then it eventually becomes pinned at some location, giving us this polarized distribution. Okay, and that occurs due to conservation. Essentially, rac switches between on and off forms, and it's not created or destroyed. And so there's a finite supply of it basically in this domain. This is however much you start with, kind of that initial bump, that's all you get to spread out throughout the system. And it's limited by the total amount. And so how we mathematically try to figure out whether or not one of our cells is going to, from a resting state, end up in a polarized state is we apply some method, which is a nonlinear stability method called local perturbation analysis that Bill is actually almost pioneered and written a lot about and used extensively in these kinds of systems. And it's perfect because it allows us to understand how the activity of rac depends on some sort of local perturbation. So you can imagine some experiment where you're perturbing part of a cell and firing up the rac activity somewhere against a sort of steady state background levels of the slowly diffusing active stuff and the quickly diffusing inactive stuff. Okay? And so the basic idea is that when you have one of these perturbations that's sort of localized, it's asymptotically small, but of very large amplitude, you can study the response of that perturbation of the system to the response of that perturbation by thinking about the different timescales in the problem. So I've just rewritten the rac PDEs just in U and V and epsilon and D, just because I'm card-carrying mathematician, as Darren said, and sometimes I have to change to U's and V's and P's and epsilons. But the basic idea here is that this first equation is the active rac, and it's got this slow diffusion. Epsilon is typically a small number. Here, the inactive rac has got this large diffusion. And if you think about what happens to this perturbation on the time scale of what really matters, which are the reaction kinetics, this perturbation basically doesn't go anywhere because it diffuses very slowly. So the effect of the epsilon diffusion on this perturbation is negligible on the time scale of the reaction kinetics. And at the same time, if this perturbation were also to affect the global V variable, on the time scale of anything happening in the reaction kinetics, it would get quickly wiped out by this fast diffusing stuff. Okay, and so basically what we're able to do with this method is exploit the fact that there's a time scale discrepancy between the two diffusion coefficients and reduce the system of partial differential equations to ordinary differential equations. And what this allows us to do is describe the system in a much more simpler way and essentially reduce the complicated problem to a rather simple one that just describes the evolution of this perturbation, which I'm calling UL, the local active perturbation. Okay? So what you can see here is that these two ODEs describe the global behavior outside the perturbation. They describe the evolution of these quantities. And this last equation describes the evolution of the local perturbation against that global background. Okay, and the great part about this method is that then you can go turn to all of dynamical systems theory and understand everything you want to know about the dynamics of this system using things like numerical bifurcation analysis. Okay, a little bit detailed here, but let me walk you through this. The basic idea is that first two equations describe the well-mixed behavior of the system. They describe what happens at the level of, if the cell had no spatial component at all, if it was just like a well-mixed beaker. Okay? And it would tell us, simulating the system or doing a steady state analysis of this system would tell us the homogeneous steady state value of Rac activity that we could expect. And that's what this black line is showing you here. So for example, if the basal activation rate was 5, we would expect a, I don't know, 1.25 or so level of Rac activity in our cell. What the local perturbation analysis does is it adds some curves to this diagram on the right, and it adds that gray curve. And in particular, this gray curve tells us information about whether or not that local perturbation will go back to the background level, or if it will grow and in doing so drive pattern formation. So for example, let's say your cell had a basal activation rate just shy of 4 or just around 4 here. Now, if you were able to apply a perturbation and increase the Rac activity there, nothing would happen because there's nothing going on up here. This homogeneous steady state is stable and the system's dynamics would return to it. But on the other hand, if you were to somehow be able to turn off Rac in that region, maybe enough to get across that dashed line or even lower, you could end up here. So the local perturbation would attain a value, a steady state value that's different than the global background level. Okay, so this means that the perturbation has grown. So some sort of pattern has formed. Right, same thing over here. If you're able to upregulate Rac locally, you get yourself across that dashed line of threshold, and then you're able to grow up to some very high level. Okay. The other thing that this method can pick out is regimes, which I'm calling a Turing regime here after Alan Turing's sort of a pattern formation analysis, is sort of diffusion-driven instabilities, is that the homogeneous steady state is linearly unstable, and so any sort of noise, small noise amplitude perturbation from it would drive pattern formation. And then you can turn to some more complicated mathematics to sort of trace out the boundary of this blue region and the boundary of this red region in higher parameter space. Oh, look, I have little pictures explaining what's going on here. Right, this is the low Rac steady state. This is the high Rac steady state. If we do indeed do that perturbation, we end up in this polarized regime where we ended up with a Rac activity profile that looks like something that's polarized, or again, a different kind of polarity where it's a little bit flatter, but nonetheless polarized. So you can take that diagram and soup it up a little bit. And now I can start to look at more of the parameters in the model to understand how the signaling responds to changes. And on the left, I'm showing you a bifurcation diagram with respect to the deactivation rate delta and the basal activation rate B. The reason I'm doing this is because I know that chemoattractant that I'm dumping on my neutrophil increases Rac signaling. It turns Rac on. So it's essentially like increasing the basal activation rate. And I also know that tension is responsible for turning Rac off. So increasing tension is like moving up on this positive y-axis and increasing the deactivation rate. So I can think about then what might happen as I move throughout this diagram with inputs from signaling and inputs from mechanics. Okay, so let me just sketch out the behavior. In this unshaded region, you have non-polar solutions. You have just the low Rac steady state, kind of corresponding to a resting cell up there at B. And that makes sense. It's a relatively low basal activation rate, relatively high deactivation rate. At E, we have a high Rac steady state Again, quite a bit of activation, very little deactivation. Inside the blue bubble, you're able to get stimulus-induced polarization, meaning with a sufficient perturbation, you can end up driving the PDEs to attain one of these polarized distributions. And inside the red bubble, noise is sufficient to attain a polarized solution. Okay, so here comes the simulations, right? Now, basically, if I do that experiment that I just described numerically and simulate the PDEs over time, you can see that I'm going to mimic a chemoattractant application by taking a resting cell and increasing its basal activation rate all of a sudden. And then I'm going to slowly let tension build up over time to push the cell back into the polarized regime. And what happens in a cell that's not going anywhere, that's just kind of a fixed endpoint between 0 and 1, the cell is initially at rest. All of a sudden, I fire up the basal activation rate B. So Rac signaling spikes bright yellow. Tension then starts to build up slowly over time until we get to this sort of max delta. That's where now our Rac system is in the polarizable regime. And just shortly thereafter, you can see the onset of a polarized solution. We have high Rac on one side of the cell and low Rac on the other. Okay, and so this is kind of just supporting our first initial evidence of our polarization induced by this sort of mechanics and chemical adaptation. So what we did next is we took this to the next level and said, okay, but what if we have a cell that's actually moving? What if we have the sort of full feedback between signaling and mechanics? And we let our Rac signal thus cause the cell to expand. Okay, so I built a very simple mechanical model of a cell. It consists of a spring coupled viscously to some substrate. And now what's going to happen is the Rac is going to move around and react inside some computational domain that's going to change size, because we're going to allow Rac to induce forces at the cell endpoints, like it would be if it was turning on actin polymerization. Okay, so if there's sufficient Rac, then there's going to be some forces that will cause the cell to expand at either endpoint. And then to couple the feedback all the way, what we do next is we take our cellular protrusions and we allow them to influence mechanosensing by increasing the deactivation rate that as I said before. So we allow the deactivation rate to increase with respect to the tension on the cell, and the tension is just basically how long the cell is minus how loud the cell wants to be. That's this L minus L naught. Okay, so we put that together, we do some work to simulate it, and what we end up getting is kind of the first result. If we don't have any feedback from tension, the cell just brightens up Rac activity everywhere with response to the uniform stimulus, and then expands a little bit over time. Okay. However, with feedback from tension, the cell expands, and as it expands, tension builds up. As tension builds up, the deactivation rate increases, and driving the signaling into the polarizable regime, where we have a high Rac activity over here, low Rac activity over here, and then the cell happily migrates on its way. One interesting model prediction is that for a different stimulus level, but the same feedback strength, you end up with this polarization-relaxation oscillation, in the sense that the cell oscillates between a sort of expanding, or once it has expanded, it polarizes, but there's insufficient Rac activity to maintain the polarity. The cell then starts to shrink just for a short amount of time. Once it starts to shrink, well, there's kind of enough average Rac activity in the cell. Since the cell is smaller, the average concentration goes up, and this then induces polarization again. And so this oscillation continues. Just going to skip a slide and jump to the kind of the coolest part of the talk, I think. And this is largely Jared's development. So Jared is an expert kind of biomedical engineer and is great at continuum mechanics. So we wanted to understand, in my 1D cell, the shape changes are isotropic. The cell basically just stretches or shrinks at the same rate. There's no shape change. There's no non-circularity, right? This is going back to Darren's question about the Legge model. How important is it that it's circular? Well, we wanted to allow and understand the influence of shape changes on our cell. And so we built this 2D mechanics model for, again, thinking about kind of the viscoelastic material that would govern cellular physics at the sort of cell surface that's adhering to the substrate. And we keep track of everything that's going on inside this domain. We keep track of the Rac activity inside the domain solved by our reaction-diffusion PDE. We know how each piece of this material displaces, and so how it moves. We know all the stresses that the material experiences, basically the forces that each material point exerts on each other. We know how Rac causes a protrusive force in the normal direction to the cell periphery. And we can keep track of the strains and whatnot. So the only real difference between this model and the 1D model is that, well, this one's a little bit more biophysically realistic because it's 2D and we've got all these great parameters from literature. And we've got this description of tension now that depends on the area minus the rest area. So I'm not really an expert at continuum mechanics, but I understand these sort of 4 sets of equations with their analogous to the 1D models. And I think I'll describe it like that to help give you an idea of what these equations are saying. This first one is basically saying that the sum of the forces inside the object must balance with the forces that it experiences from the environment, the drag that it experiences from the environment, so that it moves. U dot is basically the change in the displacements. It moves as a result of those stresses. We assume that those stresses are basically the same as the 1D model, that they decompose into an elastic and viscoelastic part, and that the elastic and viscoelastic parts basically just follow Hooke's law, and what's analogous to my little dash plots in the 1D model. Okay, and then what we can do from that is we use something called the plane stress condition to basically think of the material as just being 2D, as well as a small strain approximation here, which allows us to calculate the strain and the strain rate from the displacements. So putting that all together, getting a whole bunch of parameters from literature, such as the cell stiffness, the estimating parameters for the viscosity and the internal viscosity, the shear modulus, the force due to actin polymerization, and trying to match realistic cell migration velocities, we're able to start to simulate these cells and recapitulate the results of the experiments. So without feedback from tension, what I'm going to do is basically fire up one of these cells. It's got this dashed line showing its initial position, and I'm going to stimulate it with a high level of chemoattractant. So I'm going to increase that parameter B, and we're going to see and watch what happens without feedback from tension. So Rac activity spiked, right? It turned onto this, bright yellow level or orange, and the cell started to expand. And admittedly, it migrates a little bit. And the reason it's doing so is because we've artificially induced a very small gradient so that our cells will always migrate in the same direction. That's just a computational hack to make it easier to do data analysis. Everything is moving basically in the X direction. Now, when you do the same experiment, but you do have feedback from tension, all of a sudden you get this persistent polarity and persistent migration. And so this is, again, further evidence that this feedback from mechanics can work, provided it pushes the signaling dynamics back into this signaling domain, this polarizable domain. Okay, I just want to mention that the question of circularity and geometry and non-uniform shape changes becomes a little bit more important here. And so the polarization and relaxation oscillation that I also found in the 1D model, we can find it in this 2D model, but it's a little bit weirder. It's a little bit more transient. There seem to be some sort of, different modes of oscillation almost. The initial sort of behavior of this cell in the oscillatory regime is like a little blip, 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 blip. But as it elongates sufficiently, the oscillation sort of changes to a different kind of mode or frequency or something like that. And we get a different oscillation as the cell moves along. And all I'm trying to illustrate there is that there's some sort of different, important role for these things, geometry essentially, and the mechanics that play a role in 2D. So this is just the basic prediction of our model, is that if you can now do this experiment and go in and measure things like the cell area the rac signal, and maybe you could get tension or traction forces or something like that in one of these experiments, you could start to map out and see whether or not this tension feedback actually plays a role. And so I suspect that you could do some sort of manipulation where you are able to muck around with the cytoskeleton. I don't know exactly how this would go, but do the experiment where you could get the sort of area over time in the case where you don't have this feedback, the case where you do have this feedback, and whether you're sort of in a non-polarized state or you get a polarized state. Okay, this oscillation might also appear in experiments, I'm not sure, but that's the basic prediction that there's the change in the response of the cell with feedback from tension going from off to on. Now back to the gradient simulations. So the other fun thing we did was we stuck our cells now with their feedback mechanism into a gradient. And so in the 1D model, you can see 3 snapshots here, or trajectories. The one on the left is just a cell that just basically stays here. It doesn't go anywhere. But as feedback increases, the cells are able to migrate further up this gradient before they lose their polarity and just kind of become oversaturated. So basically what's happening, right, is the cell experiences the gradient, activates RAC on one side, polarizes, migrates in that direction. But as it experiences higher and higher levels of signaling, it becomes oversaturated everywhere and it can't go as far. The point is that this feedback can kind of act as a control system. And if you can modulate it, then maybe you can modulate how far you can get up one of these gradients. Okay, and the same thing happens in the 2D cells when you stick them in a chemoattractant gradient and you compare what happens without feedback, that's in the top, and with feedback in the bottom. You can see that the cell with feedback ends up migrating much further up the gradient, and it loses polarity much later. Okay, I'm just going to illustrate quickly how I started to visualize the polarized RAC activity in these simulations, because I wanted to sort of demonstrate that there is a change in polarity and that with feedback, there is more polarity for longer. So you can imagine, you look at the video, especially at time 300, like this snapshot down in the bottom right here, the cell without feedback kind of looks polarized, right? It's got orange to yellow. And this one's got like red to orange. So there is some gradient in both cells, and that's to be expected since these cells are both sitting in some gradient of stimuli. So we expect more on the right side of the cell than on the left. That said, it's kind of a bit of the color scheme and the visualization that maybe might lead you to think that this cell at the top does indeed look more polarized than this cell. So what I did is I imagined that my cell, each one of these cells is just like an image, and I basically looked at the RAC intensity. So that's on the x-axis here, is like the number of pixels with RAC level 1.5 or something like that, and made a histogram and plotted a kernel density estimate to give me an estimate of the probability density. And you can see over time, these 6 snapshots, what happens to the RAC activity distribution inside the cell. The blue one is without feedback, and the orange is with feedback. So the 2 cells start out at about the same point. And you can see quickly that they both polarize in response to the chemoattractant, the gradient, where the orange cell has a whole bunch of pixels at this low level of RAC and a whole bunch of pixels at this higher level. And so does the cell without feedback, right? It's got these 2 peaks in this histogram. But pretty quickly, right at about 50 seconds or so, the cell without feedback, this distribution is much more unimodal than the other one. So I'm making an argument here that this feedback is maintaining this polarized bimodal RAC activity distribution much longer than the cell without feedback. All right, let me just wrap this up. So in summary, basically what I've done today is I've coupled the reaction-diffusion model for RAC signaling dynamics and a very simple minimal model for cell mechanics, like a viscoelastic spring and dashpot system or viscoelastic continuum description of the cell. we've put together this basic circuit that RAC causes the cellular protrusions. These protrusions cause tension to increase. Tension feeds back into signaling, deactivating the RAC signal. And using the local perturbation analysis, we're able to map out the behavior in parameter space, understanding how this adaptive response could indeed work. Given any sort of overstimulation or stimulation in chemoattractant application, some concurrent increase in deactivation, like due to mechanosensing, to push the cell back into the polarizable regime can indeed cause the cell to maintain polarity. And I showed that in both 1D and 2D simulations. And basically I'm positing that these cells with feedback from tension can migrate further up the gradient than cells without the feedback before they become oversaturated and lose polarity. And so this is one potential mechanism that might work in parallel or alternative to some of these mechanical or biochemical models of adaptation. So let me just thank my collaborators again, Bill Holmes here at Vanderbilt in the Department of Physics, the great team of Jared and Vijay in the biomedical engineering department at the University of Melbourne, and a colleague, Andreas Butenschon from the University of British Columbia, who incidentally, we were working on a very similar project at the same time, and so we discussed some of the mathematics. I should also just thank the funders and point out my website and email in case there are any lingering questions or you want to get in touch. Thank you.
Thanks, Cole. That was terrific. I'm
Thanks,
always impressed by, I love it when people post a GitHub website. I'm a big fan of Git. But we have a number of questions posted, and I'll start asking them. So the first one from Philip Stouffer is it known if there is an electrical potential gradient that matches up with the RAC gradient in response to chemoattractant? I know ion channels are commonly associated with motility, but I'm not clear how that is mechanistically. I can imagine that the chemoattractant can modulate open probability of channels, thus allowing for an electrical potential gradient to be set up in the cells. This could be a very rapid, instant communication method for the cell.
Yeah, that's a great question, and I think it's totally right. So I don't, I haven't thought about that in this particular project, and we've kind of wrapped up, sort of the results that we've done here. But I will say, I think that Philip is exactly correct that things like calcium calcium ions, rushing in through different channels are moving around across the cell membrane. Definitely do, that does occur in response to environmental stimuli and in chemoattractant. The implications of it as a signaling thing, I'm not exactly sure. My guess, and this is just a kind of a gut reaction, is that the calcium signaling is very fast and very small, and these sort of gradients might exist locally, and I think that they do probably have some role to play in sort of the signaling cascade that eventually funnels down into sort of RAC activity and actin regulation. But my guess is that's on a time scale and a spatial scale that's kind of much too small to dictate and organize the cell behavior entirely. So I think that I think that's an important point, and probably is kind of one scale of the problem. We're kind of thinking of working at a certain scale here with the RAC signaling and this course level description of the mechanics. I think that there's a couple other scales of important behavior and important players. I didn't talk about actin filaments at all, even though I had that cool image in my slide at the beginning, right? So there's a scale where that stuff is very important, and I think that all of these things should be thought about sort of in an integrated theory of this migration, communication, and adaptation.
So you're essentially thinking about the input variables into RAC activity?
Yeah, that would be my gut reaction, I think. And I know that, well, I've basically had this question before. What about calcium? And I've come back to it and said, well, yeah, I think it plays an important role. And I think it's kind of at a particular scale in the signaling cascade that doesn't necessarily apply to kind of the scale and time scale that we're thinking about in this problem. But I'd be really happy to talk about that more with people that know things about calcium.
Yeah, that's directly relevant to the next question from Marina Cavarina, who asked if you could explain a little better in your model how RAC is actually becoming active? What is it that's inducing RAC activity in your simulations?
Yeah, sure. So in my simulations, I'm not actually modeling anything that chemoattractant that's turning on RAC. I'm just basically manually turning the basal activation RAC of RAC on. And so this parameter B is the only thing that I'm adjusting. I don't actually model, for example, in this moving cell simulation, I don't have like a source of chemoattractant over here at the right that's constantly building a gradient for these cells to migrate up or anything like that. All I'm doing is saying that this basal activation parameter B changes spatially and it's increasing with positive X. Okay. Now, biologically, of course, the chemoattractant, whatever it is, and there's a whole bunch of different things that go on, but basically it hits that signaling cascade, and maybe calcium plays a role, maybe it binds to receptors, and the takeaway is that there's a whole series of cascading signals that go from sort of signaling from the external environment down through other proteins, which in turn activate RAC. So there's a lot of people that have looked at the various GEFs and GAPs, that's the sort of proteins that activate and inactivate GTPases, and how other signals, such as chemoattractants, but also as signals from mechanics like integrins and other things like stress fibers muck around with the GEFs and the GAPs that then modulate the RAC and Rho signaling inside the cell. So it's a complicated and big story. In the work I've done here, it's just this basal activation rate B, which is like me turning the RAC on manually. But in reality, there'd be a whole series of signaling cascades and other things that would either modulate the RAC activity or even just bypass it and cause other things to happen.
So I had a question myself, just trying to understand how the modeling, it seems like there is essentially points within a cell that are sensing, if you will, or you're acting as the way of distinguishing what the gradient is. Is there some parameter in the model that talks about how much, what that size is compared to the cell, or however?
Yeah, so I think this is a really important question and something that we've kind of swept under the rug a little bit in our work. So you might remember I defined tension in the cell as the difference between the length of the spring and its rest length. Or in the 2D model, I just defined it as the difference of the current area versus the initial area or its resting area. Okay, so that assumes that the cell is able to somehow make that measurement, or it's able to know how different it is from when it was a little while ago or something like that. It's kind of a proxy for the whole host of important things that could go on in mechanotransduction, and there's a whole bunch of different pathways. There's some really cool work. For example, the paper title is like, "Do cell membranes jiggle like jelly or flow like jello?" or something like that. It's a really cool paper, and it's looking at whether or not the cell membrane actually gets tension gradient and whether the tension propagates all around the cell or if it's just localized. And this is a super important thing to add into this kind of modeling, because I've just assumed that it's sort of, homogeneous, a global average signal, just like the inhibitor in the Legge model, that the cell can tell, oh, I'm much longer than I was, so I'm experiencing tension, so I need to change my RAC signaling. Now, I think getting those biological details is super important, and something that we've been talking about with Jared and VJ is they have the tech basically to do this experiment, to visualize RAC, and also to try and figure out what the cellular physics should be that best describes sort of the shape changes. This is viscoelastic model, but with things like traction force microscopy, we should be able to do this experiment, kind of figure out, how much tension the cell experiences, what are the forces it's inducing on the substrate, and try and actually back out maybe a better physical model of the cell rather than using just this sort of generic viscoelastic medium. And so I think kind of both of those directions are important and necessary to kind of get at your question and try and figure out exactly what the sort of mechanotransduction elements are whether this model is okay or if it needs to be described more accurately.
Excellent. Yeah, so two more questions. We'll see if we can get to them quickly before we
Sure.
transfer over. Actually, before I do that, let me put the link to the continued discussion in the chat so that for the next hour, for people who want to continue the discussion, they can switch over. Due to the restrictions, this is the webinar format, but that format will be an easier open discussion where everyone will be able to participate verbally. So the next question was from John Vastola, who was asking, what happens if you have chemoattractants placed in competing locations, like on opposite ends of the cell?
Yeah, I don't know. I don't know exactly what would happen in this particular framework or this model. I mean, there's been a lot of work theoretically in the Leahy situation area and kind of the work that's come out of stuff from Leah Keshet's group at UBC with this wave pinning model. And, you know, people have asked these questions about how do these models and how do these cells respond to competing information? How do they respond to multiple chemoattractions? How do they respond to like a chemoattractant that turns? How should the cell turn? And yeah, there's a large body of theoretical and I think, you know, pretty well backed up with experimental work probing those responses in these kinds of models. I don't know what would happen with ours. Maybe we could try that later.
And then the last question from Venkateshwara Amara is asking, were these experiments conducted in liquid medium or a gel-based medium? And then how were the mathematical equations generated?
Yeah, I'm not sure about that. I think that's a great question. My guess is that the cells are just sitting on some substrate, and I don't know. We'd have to go back and look. The experimental group is Orion Weiner at UCSD. Basically has a whole bunch of these neutrophil experiments in different scenarios. So I'm sure those details are out there. I don't know them. The mathematical equations were generated. So the continuum mechanics model is kind of like a standard continuum mechanics model for a viscoelastic material. The simple physics-based model is kind of just like a spring and a dashpot, so you can put that together using kind of undergraduate level physics. The reaction-diffusion PDEs governing the signaling, those came about from a long sort of effort in mathematics to quantify and describe the spatial and chemical reactions seen in many sort of pattern-forming situations in biochemistry and in biology. And this particular wave pinning model came from the other group of Leah Keshet, who were basically trying to distill all of the interactions between Rac, its gaps, and feedback from things like phosphoinositides and actin and all these other players. And this sort of little circuit that I'm showing at the top right of just the on-off, switching on-off, and the positive feedback in active states is kind of like the simplest description that you can get this behavior with. And so it's kind of like a little core motif of the big signaling and governing the sort of overall dynamics of the system. So we can get away with it as like a very coarse description of all the signaling going on. So that's where the equations come from.
Excellent. All right. Well, thank you so much. And just to reiterate, we will be continuing the discussion mostly with the students at the Vanderbilt Zoom link that I placed in the chat. And thanks again, Cole.
All right. Thanks. I'll see you in a minute.
Okay.